Thank you, Albert. And I want to thank everybody for the opportunity to attend this workshop. I've learned a lot about epigenomics. I'm really going to go back home very excited about the opportunities that epigenomics offers those of us in animal science who are trying to enhance the efficiency and sustainability of animal production. I come to this whole question from the point of view of a reproductive biologist. I'm less interested in predicting biological function than in manipulating the physiology of the preimplantation embryo to increase fertility of female animals. And recently, we've been focused on manipulating the preimplantation embryo to change the phenotype of the offspring that results from that pregnancy. And that's a phenomenon that's been well characterized ever since the days of Barker. And there's a lot of evidence now that developmental programming can occur at the earliest stages of development during the preimplantation period, and even in fact during development of the gametes before fertilization. So we've been exploring this topic, and obviously this is a phenomenon that involves a lot of epigenetic regulation. This is bovine blastocyst at day 7 of pregnancy that's been labeled with an antibody to 5-methylcytosine. And I just put this in here to illustrate the fact that the preimplantation period is one that's characterized by a lot of epigenetic reprogramming. So here at the blastocyst stage is the first time in development when the embryo has undergone a differentiation event. So all of these green cells, and the green represents labeling for 5-methylcytosine, are trophectoderm cells that will give rise to the mature placenta. And then this small ball of orange cells, which label with propydium iodide, but don't label with antibody to 5-methylcytosine. Those cells are the inner cell mass that will give rise to the epiblast and eventually to the embryo and fetus proper. So there's already a lot of epigenetic reprogramming going on in the trophectoderm, but in these more pluripotent cells, the level of epigenetic or the level of DNA methylation is less reduced. And Yanni and Helen have already mentioned the fact that the preimplantation period is one where all of the epigenetic marks, or I should say, most all of the epigenetic marks of the sperm and oocyte are removed from the embryo, and then subsequently new epigenetic marks required for the embryo's own differentiation and development are placed back onto the nuclei in a lineage-specific manner. So this again is just 5-methylcytosine labeling. You can see the 2-cell embryo still has a lot of DNA methylation. That's gradually reduced, and then new methylation is acquired in a tissue-specific manner. So the preimplantation period is a time of a lot of epigenetic reprogramming. Tom Fleming at University of Southampton and other people have shown that alterations in the maternal environment during this period of development can reprogram the embryo in a way that changes the phenotype of the neonate. And these programming effects extend into postnatal life. So Tom was using the mouse, and as you can see, we're using the Brahman cow to evaluate this same question. A lot of my work has been focused on the embryokines, the cell signaling molecules produced by the oviduct and endometrium that regulate the embryo. But today I'm going to talk about another programming agent for the preimplantation embryo, and that's the micronutrient choline. So choline is a potential programming agent because of its chemistry. It contains 3 methyl groups, And depending upon what happens to choline after it enters the cell, it can get acetylated in neural cells to form acetylcholine. In most cells, it's a source of phospholipids through phosphorylation to eventually form phosphatidylcholine. And when it's oxidized, the methyl groups in choline are eventually donated to betaine, then to methionine, and S-adenosylmethionine. So choline exerts a variety of effects on the cell, one of which is to increase the availability of methyl groups for DNA methylation. And again, I know this is a really crude way of looking at DNA methylation, but that's what we did. When we used antibody to 5-methylcytosine to look for the effects of choline on DNA methylation in bovine blastocysts, you can see that adding choline to culture medium of embryos produced in vitro causes an increase in the amount of DNA methylation in the bovine blastocyst. So that led us to ask the question, does provision of choline, this is actually a mistake, we didn't actually feed choline in this study, does provision of choline to the preimplantation embryo affect the phenotype of the resultant calf? And that's an important question biologically because it can help us understand the mechanism by which developmental programming can occur. It's also an important question practically because a lot of feed manufacturers around the world are producing choline products, methionine products that have been modified to allow them to escape degradation by the rumen microorganisms so that methionine and choline, which are usually not available through the diet to cows, can be made available to the cow. So there's a lot of interest in what are the effects of choline, what are the effects of methionine on the cow, and for us on the embryo. So we did an experiment where we cultured embryos produced by in vitro fertilization in either control culture medium, this is serum-free culture medium without any choline, or we cultured the embryos in that same medium with a 1.8 millimolar concentration of choline, which is equivalent to the total sum of choline metabolites in the blood for cows fed supplemental choline. And you can see we did this in Brahman Bos indicus cows. So those embryos were then transferred into Angus recipients, pregnancy diagnosed, the calves were born, and then we followed growth of the calves until they were weaned at 7 months of age. So I'll just, here's one of our little IVF-derived Bos indicus calves with a graduate student, Eliab Estrada Cortez, who's now at the Ministry of Agriculture in Mexico, who did this experiment. So there was no effect of choline on the competence of the embryo to establish pregnancy. So in the control group, 54% of the cows got pregnant after receiving an embryo. That was 44% in the choline group. The percent that calved was 43 and 39%. Gestation length was increased for the cows that were pregnant with embryos that were treated with choline. So about 4 days longer. And you might think these gestation lengths are pretty long. Usually we think 283 days in the cow, but Bos indicus cows have a longer gestation length than Bos taurus. So choline increased gestation length. And as a result, or partly as a result, birth weight was higher in the choline-treated calves than in the control calves. And we've split these data by sex. A lot of the developmental programming phenomena are sex dependent. The effects of changing the maternal environment are different for male offspring than for female offspring. But that does not seem to be the case for choline. But nonetheless, you can see there's quite a big increase in birth weight In the choline treated calves, like 35 kilograms versus 43 in the females, 35 versus 43 in the males. If we adjust birth weight for the effects of gestation length, those effects go down, but they're still there. And then there's a tendency for a significant effect on weaning weight. And when we adjust weaning weight for the fact that not all the animals are weaned on the exact same day of age, there's a significant effect of choline treatment during the preimplantation period on weaning weight at 7 months of age. So treating the embryo with 1.8 millimolar choline from day 0 to 7 of development caused a 20 kilogram increase in weaning weight 16 months later. So, that's a pretty impressive developmental programming phenomenon and of economic importance to a beef cattle producer. So, we were pretty happy about those results, but a little skeptical. So we repeated the experiment. So in this case, again, we used Brahman embryos. We took Brahman embryo donors and then treated them in 2 different rounds of in vitro fertilization. So in the first round, half of the cows had their embryos treated with vehicle and half with choline. And then in the second round, we switched it. So every cow produced embryos that were treated with vehicle, produced embryos that were treated with choline. And then those embryos were transferred into crossbred cows, and the calves are being monitored. So this experiment's still going on. We're going to take the offspring from this experiment through slaughter. Or I should say the males are going to be slaughtered here in July, and the females we're evaluating age at puberty. But we have a lot of data already from these calves. Here's another Brahman calf with another graduate student, Lainey Heyman, who is the Ph.D. student in charge of this project. In this case, unlike the previous experiment, there was no significant effect of choline treatment on gestation length. But again, there was a tendency, just like there was in the previous experiment when we adjusted for birth weight, for the birth weights to be a little heavier in the choline-treated calves as compared to the vehicle-treated calves. Interestingly, I think very interestingly, if you're owner of a bull stud, testicular weight in the males at 4 months of age when the males were castrated was heavier for the calves that were derived from choline-treated embryos as compared to the calves derived from control embryos. You asked earlier, should you breed the younger bull or the older bull? So the younger bull is much more genetically superior to the older bull. So you want to breed the younger bull, but of course, you need sexual maturity for that to happen. So it'll be interesting to see whether this phenomenon will be repeated if we look at age at puberty in bulls. Just like in the previous experiment, we got a significant increase in the adjusted weaning weight. And in the previous experiment, it was a 20 kilogram increase. In this experiment, it was a 17 kilogram increase. So if we increase choline availability to the preimplantation bovine embryo, we increase birth weight, which producers don't like because that increases calving difficulty. But it also increased weaning weight, which producers do like, increased testes weight. And I think we've indicated that provision of this nutrient through whatever mechanism is programming preimplantation development in a way that has long-term effects on the phenotype of the offspring. So, what's the mechanism for that? That's a question that we have a lot of interest in. The obvious explanation, the one that everybody, I think, who studies developmental programming uses, is that by changing the environment of the embryo, we've changed the epigenome. Some of those epigenetic changes are inherited by the descendant cells of that embryo, so that gene expression in the postnatal animal is altered in a way that causes the postnatal changes in phenotype that we see. So, I mean, I think that's a very reasonable explanation for the kind of observations I just reported. There's a second possibility, of course, and development is a series of differentiation events, and each of those differentiation events depends to some extent on the nature of the developmental event preceding it. So an alteration in one developmental event might cause a subsequent downstream effect on later developmental phenomena to cause changes in postnatal phenotype. So maybe it's not as simple as just methylating or demethylating a bunch of genes, but maybe we're changing lineage commitment in the early embryo that has downstream effects on the nature, let's say, of muscle development in the fetus. So we want to explore both of these theories. We've not really done any experiments yet to look at, I'm trying to come up with a good name for this theory, but I haven't come up with one, the deviated developmental trajectory theory. But we do have some evidence, and we're trying to get more evidence regarding whether or not choline during the preimplantation period affects epigenetic inheritance. So in the first experiment that I reported to you with the 20 kilogram increase in weaning weight, at 4 months of age, we took a muscle biopsy of the longissimus dorsi muscle from 20 of the calves from that study, and use that muscle to do reduced representation by sulfite sequencing. We only detected 8,000 CPGs. 8.2% of those were differentially methylated between the choline calves and the control calves. And, it's a pretty small sample. But surprisingly, the major effect of choline was to cause hypomethylation of those differentially methylated cytosines. So during the preimplantation period, if you use antibody to 5-methylcytosine as your measure, which admittedly is not very good, choline causes hypermethylation, which is what you'd expect based on its chemistry. But 4 months after birth, the major result of choline was to cause hypomethylation. And a lot of those hypomethylated sites were on chromosome 27. Actually, 26% of the differential methylated cytosines were associated with 2 ribosomal RNA genes. And when we look at the association of those differentially methylated cytosines with the nearest located protein coding gene, we identified 191 genes associated with those differentially methylated cytosines. And the gene ontology terms associated with those genes are the kind of terms you'd expect to see for a phenomenon that alters muscle development, because obviously most of the differences in body weight between animals are due to differences in muscle development. So like mTOR signaling, AMPK signaling, cell growth and proliferation, cell death and survival, were all associated with those genes that were near differentially methylated cytosines. So we are also in the process of doing whole genome bisulfite sequencing on blood samples from the calves from the second choline study. We're doing that in conjunction with Marc-André Serrard at Laval University. And we have plans to repeat this experiment and look for the conservation of epigenetic marks, or I should say the conservation of changes in differentially methylated cytosines as the embryo goes from the blastocyst stage to the fetal stage to the postnatal stage. So what proportion of the cytosines that are regulated by choline in the blastocyst are retained in the fetus and in the postnatal calf? And then we have other experiments planned to use single cell RNA sequencing of the day 15 embryo to see whether or not choline is causing changes in the relative proportion of different cell lineages in the embryonic disc or in the patterns of gene expression in those individual cell lineages in the embryonic disc. So we're hoping to use this model to understand the mechanism by which developmental programming can occur. We also have a lot of interest in the practical implications of these results. So we're doing studies where we're feeding rumen-protected choline, and we have plans to do other studies where we feed rumen-protected methionine, another methyl donor, and see whether or not we can mimic these same kind of effects on postnatal growth by increasing methyl donor availability in the preimplantation period. So I just want to thank all of the students that were involved in these studies. I just showed you the two main studies. And we've also had a lot of collaborative efforts from different ranches in the state of Florida. So I thank all of them. And I'll be glad to take any questions, or one question, I guess. Thank you.
A very nice presentation. I really enjoyed. So I have one question that does the effect of choline that you see, increase birth weight, weaning weight, and the testis weight is attributed to the maternal side, like, when you see the placenta or, of the surrogate is effect, or solely due to embryonic effect, or it's a combination of both?
So, yeah, that's a very good question. And, we're treating the embryo outside the mother. So at least in this model, choline is not affecting the mother in the first 7 days of life. But then we're putting the embryo back in the cow. And of course, the embryo can modify the cow. We've looked at placental hormone secretion to see whether or not there's any, those placental hormones are regulating mammary development, regulating uterine development. So we didn't see any effect, but that doesn't mean, other hormones that we didn't measure were not affected. One thing we don't know, which I think is important, did the cows that carried the choline-produced calves produce more milk than the cows that produced the control embryos? Because one of the big determinants of growth rate will be how much milk the mothers produce. And, we were not able to measure that. So we would also like to do this in a dairy model where the calves are removed at birth so that we could look at the effects independent of any differences in milk yield due to the mother. So yeah, that's a good point.
I have a question,
Yeah.
quick one, okay? So is does parity have an influence on the
This parity of the mother
of the surrogate mothers, yeah, of the surrogate cows? Are all the same parity?
Well, I don't know. In this study, parity did not have an influence, but we've done other studies with embryo transfer where we had, like, all these cows were multiparous cows. But we've done other studies where we've looked at recipients that were in their first parity versus later parities, and then followed the dairy calves when they became cows. So in that case, a heifer calf that came from a heifer calf produced less milk than a heifer calf that came from a cow.
Yeah.
So I think uterine capacity
Yeah.
is having an effect on the development of the calf, and that affects at least its first lactation milk yield.
Yeah, that's what I was thinking, whether the effect
Yeah.
was confounded somehow.
Yeah, so I think that's an important consideration.
Thank you. Very interesting research, and it reminded me that in humans, we know that we also during development need methyl donors, and folic acid is the one that is most prominent, and also is actually complemented into food by law in the US, right? So that to make sure that pregnant women have the right amount of folic acid. So and why it was given, because it reduces the chances of neural tube defect. It actually cured, it eradicated this particular horrible disease. So then there's the opportunity to look perhaps also to see if it prevents something which is unrelated to why you're using it. And whether there is alternatives. So choline, I don't know if that works exactly the same way as folic acid.
People in human medicine have looked at supplementing pregnant women with choline, and there's some reports that it improves the baby's brain development and cognitive development. So like you can see on this slide, I work with ball chem.
Yeah.
All the ball chem scientists I know take choline every day to improve their cognitive function. But there are some reports later in pregnancy, later than I'm looking, that choline has beneficial effects on the fetus. So yeah, so it might be through a similar pathway, because it is obviously choline and the folate pathway are related to each other.